Few reminders. Last time when we had a conversation about different types of fatty acids and triglycerides, we figured that there are saturated, unsaturated, poly or mono, and the particular type trans fatty acids which are unsaturated but the double bond is in the cis position, and those trans fatty acids are actually bad for you. So you can hydrolyze, you can break down triacylglycerols, basically fat, into glycerol, this alcohol, and 3 molecules of fatty acids. It has to be done, since we're talking about hydrolysis. Water is the reagent that breaks it down, but there's got to be some catalyst. The catalyst in the lab would be acid. The catalyst in the human body would be an enzyme called lipase. Now this is more relevant for extracting energy out of triacylglycerols. Saponification, on the other hand, and it's convenient now since you literally did that 2 days ago. In saponification, you mixed fat. In your case, it was the oil, triacylglycerol in the form of olive oil. You mix it with sodium hydroxide, heated it up, and this reaction resulted in the production of glycerol and the salt of the fatty acid. These salts, the fatty acids, essentially soaps. So, I think it was Aubrey's question, are we making a hard soap or soft soap? Depending on the base that we're using. If we use sodium hydroxide, it results in a hard soap. With potassium hydroxide, a liquid soap. Now, this is the saponification reaction that you ran on Monday. So you effectively replaced water in the reaction of hydrolysis with sodium hydroxide, which produced glycerol and sodium salts of palmitic acid. So, indeed, the soaps that we use in our day-to-day life are produced by saponification of various fats. Now, I'm going to expand it a little bit because that's going to be useful for other courses such as biochemistry, biology, molecular biology, cellular biology, or microbiology. What do we use soaps for? Cleaning. To remove dirt and fat. Importantly, fat. So how does that work? Is fat soluble in water? Can we make it soluble in water? Well, kinda. Yes. So imagine you have a bit of fat, right? And you have the salt of palmitic acid, right? So you have sodium, and you have negatively charged oxygen here, 14 CH2 groups and CH3 at the end. So when you have positively charged ion and negatively charged ion, it's a salt, what happens to salts in the water solution? It dissolves and dissociates, right? So a bunch of sodium ions separately and a bunch of palmitate anions separately. Do you follow my train of thought? if something is charged, like that part of the molecule, is it considered to be, polar, hydrophilic? Yeah, if it's charged, it is hydrophilic. It can form hydrogen bonds with water molecules. So this one is hydrophilic. But when you have a bunch of carbon atoms connected to each other by covalent bonds, that part is hydrophobic. Okay, makes sense? Now, fat, hydrophobic or hydrophilic? Hydrophobic. Here's what's going to happen. You have a drop of fat in water. So this palmitate anions will surround this fat. Each of these circles represents carboxylate group, negatively charged, this group. Okay. Make sense? So this type of structures which have hydrophobic and hydrophilic parts, they are called amphiphilic. Amphi meaning two, both, or amphipathic. Those are synonyms. Okay? If you will take like a really, really greasy pan and put some water in it, you're going to have blobs of fat floating around. But if you drop a bit of a dish soap or regular soap, suddenly they disappear because soap will form those mycelium around the fat, breaking it into smaller and smaller chunks and solubilizing it. Does that make sense? You may have noticed that I never said that soap can do anything to microorganisms, because it can't. Soap is not antibacterial or antiviral. It can mechanically remove dirt, organisms, whatever you need from your skin, but it's not going to kill them. Okay? Now, let's talk about another type of lipids called phospholipids. There are a couple of types of phospholipids, glycerophospholipid and sphingolipids. So the typical glycerophospholipid contains a molecule of glycerol, okay, 2 fatty acids, not 3 like triacylglycerols, phosphate group, and the alcohol with the amino group. Okay, so we're gonna take a look at the structures. First of all, let's name some stuff here. So phosphate group here is negatively charged. Remind me, negative charge makes the part of a molecule polar or not? Polar. Now, as we're going to see, amino alcohols are also polar. We're going to figure out why. Glycerol with a bunch of carbon oxygen bonds. Is this bond polar? How do we define a polar bond? Not necessarily. So, unequal electron distribution. What determines the electron distribution along the bond? And I'm going to tell you that the electronegativity of oxygen is 3.5 and the electronegativity of carbon is 2.4. So what's the difference between 3.5 and 2.4? 1.1, which puts this bond into covalent polar. So this is polar, this is polar. Believe me right now, this is polar. Now when you see a word fatty acid, are fatty acids superpolar? Because of they have long carbons. The chain like this. What did we talk about when we mentioned, like, we have 14 carbons in line, 15, 16. Less water soluble, yes. Less polar, yes. So this is non-polar part. And the fact that phospholipids have both non-polar and polar parts make them amphiphilic molecules. Okay? But before we get to like the entire structure, let's take a look at amino alcohols. First of all, not a complete, but a bit of a digression. Remember we were talking about things like alcohols, for example. Like methyl alcohol, ethyl alcohol, propyl alcohol. How does the solubility of alcohols change when you increase the number of carbons? It becomes less soluble, right? The more carbons you have, the less polar they are, right? Cool? And the threshold, sorta, was like 5. 4 is still a little, like butanol is soluble, kinda. Pentanol already isn't. So how many carbons does this have? 5. What about this fella? There's 2. Now, the electronegativity of nitrogen is 3. Carbon is 2.4. What's the difference? 0.6. Remember, the threshold for a bond to be considered polar is 0.4. So it's above the threshold. So this is a polar bond. This is the polar bond. These are polar bonds. This is the polar bond. And these are fairly polar. So you have within each amino alcohol, give me a minute, you have not only non-polar carbon-carbon bonds, but you have a decent amount of polar bonds. So it further contributes to the polarity of this entire chunk of a molecule. Alicia, go ahead.
So the difference in electronegativity has to be above 4 for it to be polar.
0.4. Yes. Yeah, to be considered polar. And you can infer that since the difference between electronegativities between carbon and nitrogen is 0.6, but between carbon and oxygen 1.1, the bond between carbon and oxygen is more polar than the bond between carbon and nitrogen. Does that make sense to everybody? So, this is a good illustration of some phospholipids. Okay? So, does anyone have a chocolate bar near you? I'm not trying to eat it. No. Anyone has a chocolate? Nah, chocolate. Well, anyway, when you're going to have chocolate, I'll explain what to look for. So, lecithin and cephalin are 2 phospholipids. How do we know that they're phospholipids? So this is the lipid part. Okay? Right? Non-polar. Okay, if you look at this part, these bonds, fairly polar, right? this negatively charged is really polar. And choline and ethanol amine are, fairly polar molecules as well. Do follow my kind of logic. So the part that I just shaded is nonpolar. The part that I didn't shade is polar. You can represent the phospholipid. I don't like this images. So you can represent a phospholipid crudely, I admit, as a polar head and two nonpolar fatty acid tails. Making sense? So, indeed, these guys are found in different tissues. But the reason why I asked about chocolate bar, next time you eat chocolate, please read the ingredients. You're going to find lecithin there. Okay? Now, why? Lecithin. Why would you add a molecule that specifically has a polar head and two nonpolar tails to chocolate when you prepare it? Full disclaimer, lecithin in chocolate is not derived from human brain or nervous tissue. It is plant-derived lecithin. But why would you need that? We're going into food chemistry. Is chocolate fatty? Oh, very much so. Actually, the more cocoa butter you add to chocolate, the better it tastes. And cheap ones will substitute cocoa butter with things like palm oil. Okay? Cheap fat. Good? One of the reasons you need to do that, well, first of all, the cocoa itself, when you kind of crush it and prepare it to become chocolate, there's a lot of cocoa butter, and there's a lot of theobromine, which is the stuff in chocolate that is a stimulant that makes you feel great. Like it really is kind of a quote-unquote narcotic, similar to caffeine, for instance, okay? Now, cocoa butter is fat. You have to mix it with things like sugar and sometimes milk, which aren't really that fatty and water-based. So how do you mix them well so that the cocoa butter is distributed within that watery substance? You add something that will make those micelles that I drew over here, in this case, lecithin, because it is absolutely safe to consume. Lecithin in chocolate, for example, and other products, it is what we call a solubilizer. Okay? Does that make sense? Anything that I can't think from top of my head about anything besides sweets, okay, but anywhere where you have to add substantial amount of fat and make sure that it doesn't form blobs. Am I making sense? You would add it. Actually, what's going to happen if you would put chocolate in a fridge and leave it there for an extended period of time? It's going to be covered with like white, I don't know, pellicle. At the colder temperatures, fat and water, the lecithin does not work as well. You see what I'm saying? It doesn't solubilize fat as well, and the fatty part of chocolate and non-fatty part of chocolate starts separating. And the fatty part ascends to the surface of the bar, and basically it's still the same chocolate, just separated, and it's not going to taste as good. That's it.
When you leave like meats and stuff in the refrigerator and the fat separates and
Not meats, well, dairy. If you leave raw milk, I'm not condoning consumption of raw milk, but if you collect raw milk and put it in the fridge, the fat will get on the surface.
or like I meant like a pot roast kind of thing, like it's
Yes,
fat and like that orange fat.
Yes.
all separated.
yes, that's fat that starts floating. It clumps together and floats on the surface. Okay, it's going to happen even if you leave whatever the pot roast sauce on the counter. Well, it is going to go sour, but before that, fat will start ascending to the surface because there is nothing to homogenize it, nothing to solubilize it in water. Okay, so this is what we just discussed, that glycerophospholipids have a polar and nonpolar regions. And because of that, nonpolar region can interact with nonpolar substances, and polar region can interact with polar substances, and we are mostly interested in water. So the reason why we talk so much about phospholipids is that they form cellular membranes. Because if you think about glycerophospholipids are indeed the most abundant lipids in membranes. And if you think about the task that membrane has to perform, it has to separate the outside and the inside of the cell. What is the most common chemical compound found in the human body? Humans, about 60% water. There's water outside of a cell, there's water inside of a cell, right? How do you separate two compartments that are filled with water? Let me ask you this. Sugar, is it water soluble? So if you need to separate two water compartments, would you build the wall out of sugar? No, because it will be dissolved. So you can't separate two waters with something that is water soluble or hydrophilic, because it will be dissolved. So you need to separate it by something that is hydrophobic, but you can't, because hydrophobic compounds will not interact, will not form stable structures in water. This is why when a cellular membrane is formed, yes, you have polar heads. This is annoying. You have polar heads sticking themselves to the surface and hydrophobic tails facing the inside of a membrane. So you have water on both sides. These are hydrophilic. Well, actually, the surface of a membrane is hydrophilic, but the actual job is done by that hydrophobic layer of fatty acid tails. Does that make sense? In this illustration that you see on the slide, the polar head consists of a phosphate and the amino alcohol, and the tail, in some cases, this part is included. So glycerol is included in the polar head. But fatty acids are definitely nonpolar and form those two polar tails. Does that make sense to everybody here? Good? Okay. So now, this snake, I told you before, like chemists should ask biologists about the details like this. Are all snakes the same? I know. Is there more than one species of venomous snake? Do you think their venoms act differently? Probably, yes. So it is true that in many snakes, what happens is the venom, when it gets in the bloodstream, breaks down red blood cells, which is not great because it will, first of all, cause hypoxia, then it will cause kidneys to shut down, and eventually a person dies of acute kidney failure. Okay? But not all venom snakes work like that. Okay? For instance, venoms that are derived from, broadly speaking, vipers have a completely different mechanism, which is very exciting, but has no relation to phospholipids whatsoever. Okay? We can, if you want, we can chat about it after the class, but now we're going to move on to... I just want to, when you look at this, when it pertains to chemistry, sure. When these folks talk about biology or, God forbid, medicine, take it with an enormous grain of salt. Okay? Sphingomyelin is a different type of phospholipid. It's still phospholipid. Okay? But instead of glycerol, there is a different residue called sphingosine. It also has amino alcohol. So sphingosine is an amine. Now, hear me out. I'm going to do like the part of it. So, this chunk of a molecule comes from glycerol. Agreed? Hang on a second. I'm going to go back and I'm going to illustrate my point. See that? Comes from the glycerol, right? Now, the adjacent chunk of a molecule here, those are fatty acids, right? Are fatty acids carboxylic acids? Do they have a carboxyl group? Yeah. Look, we are super fast pacing through this. So this is it's real. I understand how hard it is to retain the information and get back to it when we covered it like 2 weeks ago, just 2 weeks ago. So remember, this is the carboxylic group. When you combine, say it again, it is an ester. So when you combine a carboxylic acid, which is a fatty acid, okay, and an alcohol, which is glycerol in this case, you're going to get an ester. Right? We just talked about it. So, okay. So these bonds right here are ester bonds. Sorry, I had to include, like, it's too small of an image. Like, these are ester oxygens. They form an ester. Now remember, if you take, instead of an alcohol in this scheme of things, if you take an amine, so plus amine, you're going to get an amide. Okay? So instead of oxygen here, you're going to have the NH group there. So you're going to have CH2 NHCO, and then whatever fatty tail you have. Making sense? I'm trying to illustrate what happens here. What is an amide bond? Okay? Good? So, let's take a look at sphingomyelin. This is the residue of palmitic acid. Okay? This amide group. Sounds good? This was an OH, but not anymore. So, it's not 1 out of 10, but 2 out of 10 question. This part, when it used to be a CH2OH, what organic group does it represent? Alcohol. Phosphate. What is it derived from? Phosphate. So when you see that group, so PO4 3 minus, where does it come from? Like, I can't say, I can't give you any hints because I don't have hints. Like phosphate, which other or inorganic compound you can pair it with? Phosphates can be produced from phosphoric acid. So phosphate comes from an acid. When you combine an alcohol and an acid, what do you get? The answer is on the screen, written. No. Alcohol and an acid. Alcohol. Ester. Does that make sense? So, here's the rule of thumb. We used to talk about esters as the combination of organic acid, right? Carboxylic acid. And an alcohol. Do you follow my train of thought? But actually, if you combine an alcohol and inorganic acid, you're also going to get an ester. Just going to be a weird-ass ester, but this bond between an alcohol residue and an acid, this is an ester bond. Am I making sense? So, sphingomyelin contains 2 different types of bonds. It contains an amide bond or group between an amine and an acid, and ester bond between an alcohol and an acid. Am I making sense? Okay. So sphingomyelin, as you can imagine, because of the charged phosphate group, because of the amide group here, and because of that positively charged quaternary amine, wherever you see the charge, there is some polarity involved. Sphingomyelin is also an amphiphilic molecule, so it has a polar head, nonpolar tail. That's your, actually 2 nonpolar tails. This is nonpolar, it's pretty long as you can see. And this is it gets incredibly messy, so since all of it is recorded, I'm not particularly concerned that I'm erasing this. So what I'm going to do, we're going to highlight what's polar and what's non-polar. Sounds good? So the entire chunk here and the entire chunk here are non-polars. Yes. Okay, technically speaking, this part, 3 carbons over here, are considered non-polar too. I don't include them in the consideration because they kind of are connectors. You say okay, whatever. It gets a little tricky. Now, this, and this, it's pretty polar. Okay? Reasonable? Awesome. So now we're taking like a 90 degrees turn, not 180. Hear me out. We're still talking about lipids. We're still talking about compounds that are, I have a brain fart, I'm sorry, hydrophobic, but the structure of cholesterol, like steroids, doesn't look like the fats in any way, okay? Like a traditional fats. So this is a steroid nucleus. Remind me, please, I don't remember if you need to know the numbering system, but I seriously doubt it, okay? Does that make sense? I seriously doubt that you need to know the numbering system for steroid nucleus. Okay? Basically, what you have here are 3 6-member rings, right? A, B, and C. See that? And then you have 5-member ring that is D. No double bonds yet. There may be some. But important, this is basically in this corner, you have a carbon which is connected to 2 hydrogens and 2 more carbons, right? Am I making sense? Do you understand what I'm trying to say? So a bunch of carbons connected to other carbons and to hydrogens. Carbon-carbon bond, is it polar? Bond bit, no. Carbon-hydrogen bond. Electronegativities, carbon 2.4, hydrogen 2.1, which is less than 0.4, so it's considered to be nonpolar. Right? Make sense? So, this whole thing is extremely nonpolar. Even if you throw a couple more methyl groups, it's not going to change anything. So this is cholesterol. Okay? Extremely nonpolar. As a matter of fact, our lab next week, you're going to be isolating cholesterol from the yolk, the egg yolk, which has a buttload of cholesterol. Okay? Now, cholesterol is this part of cholesterol, as you can imagine, the part that contains carbons and hydrogens only, is very much nonpolar. And this small bit gives a tiny little bit of polarity. But not much, to be honest. So now we're going to debunk another myth about food. This is going to be a myth that will make you feel better. Okay? So this is true. Cholesterol comes in our diet through dairy products. Butter is pretty heavy on it. different types of meats. Pork is more loaded than beef. I don't know about lamb though, but whatever. And eggs. Eggs have a lot of cholesterol. And then cholesterol is also synthesized in the liver, for example, from citric acid. There's a pathway. What is the percentage? Like, what percentage is synthesized in the liver and what percentage comes from the diet? You don't have to know that. I'm asking, like, give me your guess. What do you think? Okay. Which of these two sources is responsible for more cholesterol? Food or synthesis in the liver? So, people who sell you food want you to think that it's food. 15%, 85%. Okay? That make sense? So, do you think that if you will eat cholesterol, I mean cholesterol-rich products, I'm not arguing for like pure, do you think it will alter your blood cholesterol levels a lot or a little? A little, because it's just 15%. Go on. Go on.
Does it depend on the food? Because
Mm-hmm.
there's different kinds of cholesterol.
Yes,
And eggs are healthy cholesterol.
you are absolutely correct. You're 100% correct. Now, we're gonna talk about this. We're gonna talk about. Let me just go through this and just say, okay, this statement is correct. Period. It is correct. Okay? That make sense? This statement. Not really. Kinda BS. Okay? We're going to get back to that statement. Because there are some bits in the statement that are kind of right, but altogether it is BS. Because cholesterol per se, itself, does not clog the arteries. The mechanism is different. Cholesterol does have a role in it, and we're going to chat about it.
Are foods that are high in saturated fat much more dangerous than foods that are high in cholesterol?
Let me get back to this. All right? Let me get back to this. This is one of my favorite topics. So, this statement is correct and useless. Because medical community, remember, like the lines that we draw, the levels that we consider good or bad, are a consensus and they're somewhat artificial. Not that they like bullshit, it's just where everybody agrees, we notice that if people have consistently, I don't know, blood glucose over 100, it's not good. They're going to go into, diabetes eventually. So this is kind of true as well. And we discussed that it is obtained from foods and synthesized in the liver. That shows you the amount of cholesterol in different foods. Okay? That makes sense? But here's the question. What about this good and bad cholesterol? Cholesterol itself isn't good or bad. It's like molecule of water is not good or bad. It is its combination with other fats. So I'm going to present to you several bits of evidence, very straightforward, great topic for an argument at the dinner table during Thanksgiving instead of talking about politics. People will hate you just equally. Okay? So, Exhibit 1. It has been demonstrated, literally demonstrated, that the consumption of food such as meat, dairy, or eggs in the diet does not result in the increased risk of cardiovascular disease and doesn't lead to increased blood cholesterol. Yes, exactly. When I say consumption of those foods, I'm being reasonable. It doesn't mean that you should switch to a diet that consists of pork and butter and egg yolk exclusively. Not a great idea, but the dangers of those foods are extremely over-exaggerated. What does increase the cholesterol levels in the blood more dramatically are trans fats. But it turns out that the total cholesterol levels are not predictive of cardiovascular disease. So if my total cholesterol is 220, it pretty much means nothing. I mean, if it's 1,000, probably there is some kind of serious trouble, right? But 220 means nothing. Here's what we need to do. Now, we're going to dive in a little bit of biochemistry. When you consume fat, fat has to be transported in the blood because blood is watery, right? It is transported as lipoproteins, okay? When you eat stuff, like a lot of fatty stuff, fat is loaded on proteins in the liver. And it is shipped to the tissues in the form of low density and very low density lipoproteins. Are you with me? Why do tissues need fat? Huh? Now, that's like fat, like adipose tissue. What I'm saying, like molecules. What is the role of triglycerides? What do they provide your tissues with? The E word. Energy. Folks, energy. They are better at, we were talking about this, the caloric content of food. Fatty foods are high in calories, right? And if you're active, your tissues will use almost all the fat and will ship it back. Well, they're going to not use all of it, but some of it will be shipped back in the form of high-density lipoproteins. Less fat, more proteins in those complexes, okay? If you live a healthy lifestyle, and by healthy lifestyle, I mean walking, exercising, being active, then your tissues will use more LDL, producing more HDL. Okay? This is an ideal picture. This is what you want. And when you look at your blood biochemistry, you need to look at the ratio between HDL and LDL. Does that make sense? You want more HDL, less LDL. Even if your total cholesterol is elevated, it means nothing. Because, another bit about plaques, it's not cholesterol that clogs the arteries. It's the cholesterol that some cells in your blood vessels consume, and they prefer these guys. They will consume the fat. That triggers certain biochemical changes in the cells, and the cells become inflammatory. And that inflammation in the vessel results in the growth of the plaque, composed mostly of other cells. Am I making sense? Awesome. And here's the final nail into the whole idea that the total amount of cholesterol in your blood is associated with cardiovascular disease. We're getting into medicine. Sorry, but it's relevant. You can talk to your relatives and just ruin their days. Have you heard about statins? The drugs called statins. Very successful drugs. A bunch of money is made on them. If you have an elderly relative with high blood pressure, they usually take statins. For a very long time, it was thought that statins fight high blood pressure because they lower blood cholesterol. They do, and therefore lower those plaques and blood vessels are better, pressure is good. Making sense, everyone? Well, if you think that lowering lipids will improve blood pressure, you should try other compounds to lower lipids. And it was done. And a bunch of other compounds successfully lowered cholesterol in the blood, but did not change the blood pressure. So the point that I'm trying to make is that if your lipid profile has normal high HDL, low LDL, and maybe elevated cholesterol, there's nothing to worry about. All the evidence that we have doesn't provide a definitive link between consumption of cholesterol-rich foods and cardiovascular disease. Consumption of trans fats, on the other hand, does have a link. Okay?
It's just crazy when you see the serving sizes up on the page.
Oh, it's un-American, sorry.
Like for 3 ounces of like chicken or fish.
I'm telling you, it's un-American. There should be added in front of each of these numbers, because the American serving size should be like 11 cups of whole milk, 2-pound steak. It's.
Well, it's like even if you go to even if you get like the smallest like filet mignon, like a petite filet, even that's going to be at least like around 6 ounces.
Yes, I know. Food size in American restaurants blows minds of Europeans completely. And the fact that somebody can consume it is the second blow that delivers a knockout to this. Okay. Bile salts. Synthesized in the liver, stored in gallbladder and concentrated in gallbladder. They are helping in absorption of all the fats. So this is a bile salt here. See that? You have a polar part. You have a nonpolar part. Amphiphilic molecule, folks. This part will bind to fats. This part will be exposed to watery environment of your gut. Okay? Now, this is what we were talking about. Lipid. I'm going to go back to bile salts. I'm going to tell you a little story, because I want to finish this today. So, lipoproteins, as we mentioned, participate in lipid transport. What happens is cholesterol forms, remember, there should be an H group here. So this oxygen gets deprotonated and forms the bond with a fatty acid. So you have an ester, effectively, between the cholesterol, that's technically an alcohol, and the fatty acid. Am I making sense? Okay. Then you load up your proteins, okay, with a whole bunch of fat. So when we talk about lipoproteins, okay, what actually we see here is that you have a layer of phospholipids on the surface. See that? With some cholesterol molecules and with some lipids. Lipids and the polar heads are exposed to the outside and can interact with water in the gut. Inside this complicated, pretty much mycel, you have a whole bunch of cholesterol esters and triacylglycerols, or oils and fats. Okay? So this is what I was talking about. Let me ask you a question. Fat in water, what floats? So it means that fat density is less than the density of water. Look at the densities. Chylomicrons that are formed when you just consume fat have the lowest density. They have the greatest percentage of fats in them. 2%. See that?
So like LDLs would be considered, or no, so LDL is a bad cholesterol, HDL is a good.
Yes.
But low density, so would LDLs be less dense with fat?
Yes. Yes. So look at the percentage of protein. It progressively increases. See that? So since it increases, the amount of fat decreases, the density goes up. Chylomicrons are traveling from the intestines to the liver. That's it. That's the final destination. No matter what fatty food you eat, that's going to happen. Liver decides how much LDL VLDL it makes, and your tissues decide how much HDL is returned back to the liver. Okay? So this shows you that LDL transport fats from liver to tissues, in this case, like muscles and heart, and HDL transported back from the tissues to the liver. Now, steroid hormones, all you need to know that they are, exist. Okay? They are required, and they are made of cholesterol. Okay? The most famous, of course, male and female sex hormones. Okay? Testosterone, progesterone, estrogen. And also the hormones of adrenal cortex, such as no, they are amines. Adrenal cortex, norepinephrine, it's adrenal medulla. So for instance, cortisol, the stress hormone, okay? Aldosterone is the mineralocorticoid that regulates the sodium balance. I'm not sure why we're even talking about this. I don't see how it is relevant to any of your experiences. Basically to illustrate that they are similar and are derived from cholesterol. So, let's see how far we are. Okay. So, I'm going to go through this. So, mention that there are anabolic steroids. The reason why we want derivatives is because testosterone is not well tolerated by the liver. Talk about it. Yeah, they have a bunch of side effects, but the best modern ones, they much better. Like, I think oxandrolone and androlone are pretty good. I'm not, again, encouraging you to use anabolic steroids for muscle mass, but they do promote muscle growth. Okay? Now, these two corticosteroids we mentioned, they are produced in the adrenal cortex, in adrenal glands. Aldosterone regulates sodium and water balance, and cortisol produces stress-related responses, mostly by increasing blood glucose. So those are, one of the effects of cortisol is that it has an immunosuppressive function. So if you would compare the cortisol and prednisone structure, they are similar to some extent. And prednisone is what? When do you receive prednisone? To treat what? Inflammation. You receive prednisone to treat inflammation. Now the membranes. So we're going to mention a few things about the membranes and wrap it up. So we have all the foundations to talk about it. We did discuss that membranes consist of phospholipids. We did mention that they can incorporate proteins. And we did talk about the fact that they separate watery intracellular environment from the watery extracellular environment. Since they're made out of phospholipids, which are amphiphilic molecules. Phospholipids form lipid bilayer. Polar heads stick on the outer and inner surfaces or leaflets of the membrane, and nonpolar tails are on the inside, as you can see from this illustration. So the green spheres represent the polar heads. The tails are inside. Now, membrane isn't solid, and the current model of a membrane is a fluid mosaic. What does that mean? It means that molecules that are embedded in the membrane can move around. Okay? So, these proteins can actually move, and so can cholesterol molecules. Okay? And phospholipids, obviously. So, the reason why phospholipid bilayer is kind of a very viscous liquid is due to the structure of each phospholipid. So this is polar head, and these are nonpolar tails. So do you remember which type of fatty acids has a kink? Is bent? What do they have to have that kink? Huh? Yes. So, how do we call fatty acids with a double bond? Yep. So, you can imagine that these bent molecules will prevent phospholipids to be stacked densely. Okay? You follow me? So here's my question. Imagine you create a membrane in which phospholipids have only saturated fatty acid tails. And then you create another membrane in which half phospholipids have saturated and unsaturated tails. Which membrane will be more fluid? The one with unsaturated? Huh? Of course. So cells can regulate the fluidity of membranes by preferentially incorporate saturated versus unsaturated. More saturated, more solid, can withstand greater temperatures. More unsaturated, more liquid, can withstand lower temperatures. Final bits going to concern the transport. So there's a diffusion. Now, facilitated transport is technically also a diffusion. This is simple diffusion. And then there's active transport. So in diffusion, whether it's passive, simple, or it's facilitated, you don't spend any energy. In active transport, though, you do, because you move ions against the gradient, which is pretty well illustrated here. So look at this. These molecules, they travel straight through the membrane. So, simple diffusion. These molecules have to use a channel. They still go from high to low concentration. Facilitated transport. These molecules go from low to high concentration, so active transport, which requires energy. Okay? Sounds good? Okay, awesome. So that concludes our lipids. Next week we're going to talk about amino acids, I think.